Hello, today I'm going to be talking about Helicobacter pylori. So just a little overview. It was discovered in 1982. It is a spiral-shaped microaerophilic bacterium. 30 to 50% of the population in the developed world become infected with this. It is a carcinogenic bacterium. That means it can cause cancers. It's the principal cause of chronic gastritis, gastric ulcers, duodenal ulcers, gastric adenocarcinoma, and gastric mucosa-associated lymphoid tissue lymphoma. It's associated with extragastroduodenal diseases as well. So Barry J. Marshall is the man that discovered this. He was studying curved bacteria at the hospital where he was training in cardiology, actually. A different physician wanted him to look into these biopsies that were showing this bacteria in patients with ulcers. At the time, gastric ulcers were accounted to stress, and patients were actually prescribed an antidepressant for them. Marshall was unsuccessful in developing an animal model, so he decided to experiment upon himself. In 1984, following a baseline endoscopy which showed a normal gastric mucosa, he drank a culture of the organism. 3 days later, he developed nausea and achlorhydria. Vomiting occurred, and on day 8, a repeat endoscopy and biopsy showed marked gastritis and a positive H. pylori culture. At day 14, a third endoscopy was performed, and he then began treatment with antibiotics and bismuth. He recovered promptly and thus had fulfilled Koch's postulates for the role of H. pylori in gastritis. So the taxonomy for this, the kingdom is bacteria, subkingdom Negibacteria, phylum Proteobacteria, class Epsilon Proteobacteria, order Campylobacteriales, family Helicobacteriaceae, genus Helicobacter, species Helicobacter pylori, also known as H. pylori. Transmission, epidemiology, and prevention. So the transmission is thought to be fecal to oral or oral to oral transmission. 30 to 50% of the world population is infected, and it's more prevalent in developing areas. Most people are actually asymptomatic with this. Stomach ulcers may form, however, in about 15% of people, which could also lead to bleeding, and about 1% end up forming an adenocarcinoma from this. So ways to lower your risk include practicing good hygiene. So drinking clean water, washing hands well before eating, washing hands well after using the bathroom. Anatomy. So H. pylori is a gram-negative bacterium. It possesses a unique lipopolysaccharide structure. The LPS has a strong immunostimulatory and inflammatory capacity, and it contains about 4 to 6 unipolar flagella. The anatomy that I've listed here with the LPS, that is going to come up later for how they're actually able to infect. So the stomach physiology. So a human stomach, the stomach mucosa contains stomach acid secreting cells. It also has cells called goblet cells that produce a mucus that protects the stomach lining from digestion. So here you have that layer. You have the mucus cells that create that mucus layer. So you can kind of see the mucus layer here. And then up at the top in the stomach lumen is where it's the pH2. You can't have that pH2 down by our cells, or else it would just start digesting itself. So the H. pylori response. So H. pylori is able to survive at low pH, which is why it can infect the stomach. Most bacteria are not able to survive. It does produce large amounts of an especially efficient urease. It converts urea, which is found in the bloodstream, to an alkaline compound, ammonia. What this does is it raises the pH around it so that it can survive in the acidic environment of the stomach. The mucus layer disruption then, so it also then will enter the cells of the lining of the stomach, and it creates a disruption, which then causes gastritis, this inflammation as the immune system is responding. So there are 3 outcomes from your immune response. The inflammation in the response eradicate the bacteria. It's a mild inflammation that does not damage the host or the bacteria, and they end up living symbiotically. Or the inflammation does not rid the body of the bacteria, but chronic inflammation ensues, which leads to tissue damage, which is that ulceration, which can cause bleeding. So here we just have different images of what the gastric epithelial cells look like. So the first one is a healthy, uninfected person. You have the epithelial cells, their resting condition, there's no inflammation at all. You have an intact basement membrane, the submucosa, gastric lumen, and then you have a H. pylori coming in this acute inflammation. So here your body detects it. It's starting the recruitment of the immune cells for the bacterial clearance. We have some pro-inflammatory cytokines and initiation of gastritis. After a while, though, what that leads to, if your body can't get rid of it, is a chronic inflammation. Now, this is when you're going to start to see these negative side effects from it. The immune cell accumulation due to this prolonged infection, if untreated, it can progress to an adenocarcinoma. It also can cause that ulceration. And then the last stage, which is gastric cancer. So for this, you see that the basement membrane that you see in the other 3 slides is no longer intact. The cells are kind of uncontrolled and just taking over here. So virulence. So 85% of the infected patients only have mild asymptomatic gastritis, while 15% can develop to have an ulcer, and less than 1% actually develop to have gastric cancer. There is a diverse clinical presentation of H. pylori infection, which is a result of the interaction between the bacterial virulence, the host genetics, and environmental factors. The virulence factors responsible for establishing colonization include urease, flagella, chemotaxis system, and adhesions. So there are 3 major pathogenic processes, which is colonization, the immune escape, and then disease progression. So for colonization, we have urease, which neutralizes the gastric acids. We have flagella chemotaxis system. What that does, it allows the H. pylori to have movement to the epithelial lining. And then you have adhesions, which are what kind of hold it to the gastric epithelial cell. So immune escape. There's a lot of different ways that it escapes the immune system. One of the ways is it alters the PAMPs. What this allows is to avoid the toll-like receptors. They're looking for specific PAMPs on the surface, but there aren't very many, and the ones that are there are adjusted so that they don't get detected. There's also something called VACAE, GGT, and arginase. What those all do is they inhibit the T cell proliferation. The H. pylori OIPA decreases the IL-10 levels and suppresses the dendritic cell maturation. Here's that VACAE again. It's a secreted pore-forming toxin that will enter the host epithelial cells by endocytosis and affects a variety of biological processes. And CAGA also enters the cell and causes polarity disruption. So this all impacts your immune system not being able to properly respond. And disease induction. So a few different ways it can go with disease induction. You have gastroadenocarcinoma, you have the peptic ulcer disease, PUD, or you have duodenal ulcers as well. So gastric adenocarcinoma and PUD. So like I said earlier, the CAG-A, which is cytotoxin-associated gene A, it is a oncoprotein. It is injected into the host cell using a pilus structure, which is called type 4 secretion system, or T4SS. So this bonds with the host cell and is able to pass this along. The CAG-A alters the intracellular signal transduction pathways that facilitates malignant transformation of gastric epithelial cells. The increased gastric inflammation via the NF-κB signaling and increased IL-8 secretion. And CAG-A can also cause epigenetic changes such as DNA promoter or histone hypermethylation, which in turn leads to the downregulation of tumor suppressor genes. So your genes aren't acting on these cells that are kind of out of control replicating. And then we also had the vacuole cytotoxin A and the blood group antigen binding adhesion, which is a BAB-A. Those three really are the factors that contribute to the gastric adenocarcinoma and PUD. PUD is also, other than those first three I mentioned, the CAG-A, the VAC-A, the BAB-A, duodenal ulcer promoting gene, DUP-A. It's a neutrophil infiltration and IL-8 expression in the antrum section of the stomach. Induced by contact with epithelium gene A, which is the name, ICE-A, increases mucosal levels of IL-8, which IL-8 is used for inflammation. And outer inflammatory protein A, higher H. pylori colonization density is associated with this, neutrophil infiltration, and IL-8 levels in the humic stomach. There's also a range of extragastroduodenal diseases that are associated with this. The first one would be iron deficiency anemia. So with this, the iron loss could be attributed to a few different things. It could be attributed to the hemorrhagic gastritis. So those ulcers are causing bleeding, you're losing blood, which means your iron is going down. The peptic ulcer disease, gastric adenocarcinoma, they're all caused from H. pylori, and they all can cause this bleeding, which causes iron lower. CAG-A protein enhances the iron uptake during the growth of the bacteria. H. pylori-related corporal gastritis. So what this can do is it decreases acid secretion due to gland atrophy, which can result in reduction of iron absorption from the diet because you don't have the acid secretion to help with the digestion to get these nutrients out. And this correlation is important for treatment because incorporating the H. pylori treatments along with the iron supplements is really what's going to help to get people back healthy. Other extragastroduodenal diseases that are associated: malabsorption of vitamin B, metabolic syndrome and diabetes mellitus, immune thrombocytopenia purpura, non-alcoholic fatty liver disease, coronary artery disease, cerebrovascular disease. So testing for H. pylori. So there is a non-invasive way, which is a nuclear medicine imaging scan. Urea is labeled with a carbon isotope, either carbon-13 or carbon-14, and it is then ingested by the patient. So when this urea is labeled urea reaches the stomach, the H. pylori breaks it down with the urease. This radioactive carbon then becomes a CO2 byproduct that is transported to the lungs through the bloodstream to be breathed out. So actually what they do is they take a breath sample from the patient that can measure the amount of radioactive material to determine if the CO2 is radioactive. Other ways for testing for H. pylori, a biopsy. So this is mostly done through endoscopy. A flexible tube is inserted down the esophagus, down into the stomach, and they can take a tissue sample to test. They also look for signs of the H. pylori in the stools. They also can do serum testing. The problem with the serum testing, though, is that it doesn't really show active infections as well. So if they've gone through treatment, you can't really tell as well if it got rid of the H. pylori. It just still shows that they have had H. pylori recently. So treatment. Treatment is usually a combination of 2 to 3 antibiotics that are taken along with a proton pump inhibitor for periods ranging from 3 to 14 days. You can also use antimicrobial drugs and Pepto-Bismol. And that is it. I hope you learned a little bit about H. pylori.